2 more minutes. But as I can see, people are kind of jumping on at a rapid rate. We'll just give a couple more minutes before we get going. Thanks very much. All right, I'm back. I think given that we've got a full jam-packed agenda, we will get going. So thank you all to those who are joining right now. We are also, we're recording this, and B, it's being live streamed on our YouTube channel as well. There we go, we've started recording. So welcome everyone, it's our first episode of our Molecule to Medicine series. The episode is called The Drug Invention Process. So as a bit of context, Cyclica has been hosting educational webinars for a number of years. It was formerly known as AI Ate My Homework, and with the original objective to help and educate students within the drug discovery process. Our following grew to quite an array of people, and ranging from students to academics to potential investors to people within the biotech community and I guess in general pharma industry folk. But at our core, our aim is to give back to the educational community within the realm of drug discovery. So before I introduce the episode, set the stage for the episode and the speakers, we must first acknowledge the land we are meeting on is the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, and the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis peoples. We also acknowledge that Toronto is covered by Treaty 13 with the Mississaugas of the Credit. All right, so I will just move along to the next slide here just to give you the full picture of what we have in store for you for the Molecule to Medicine series. So right now we're on episode 1. As you can see, the general topic is how molecules become drugs. So I'll introduce the speakers, Andrew and Vern, who will take you through that entire journey. But just so everyone can see, over the next few months, we're going to cover these additional topics, and you'll see that the entire roadmap amounts to learning about how a molecule becomes a medicine. So that's the objective of our series. So if you have any questions, you can always go to the Molecule to Medicine website. You can ask questions, register for events. It's within the Cyclica website, so you can find it there. Now, without further ado, I'd love to introduce our 2 fantastic speakers for today. So meet Andrew. So Dr. Andrew Fowler completed his PhD at Southampton University and went on to work for Beecham Pharmaceuticals for the next 15 years, which is now known as the small pharma giant GSK. Andy was a project and group leader at GSK before moving to Eli Lilly in 2003 to become head of chemistry at the Windlesham site and remained there until the site was closed in 2020. Currently, he is CEO of Slowwave Therapeutics, a new biotech company focused on using slow wave sleep to improve brain health and combat dementia. During his career, Andy has worked on asthma, inflammation, and diabetes targets, but his main focus over the past 20 years has been in neurodegeneration and in particular, Alzheimer's disease. So welcome, Andrew, and thanks so much for joining today. And we have our very own Dr. Vern de Biasi. Vern is the Chief Partnership Officer at Cyclica and supports Cyclica's growth strategy by scaling Cyclica's partnerships efforts and global presence. Prior to joining Cyclica, Vern served in multiple senior leadership roles with over 20 years of experience in discovery and late-stage development at GSK. Vern has global experience, having led teams in Italy, China, the UK, and the US. Vern received his PhD in analytical chemistry from the University of Hertfordshire. Just before we move on, Vern will take the next slide here. I just wanted to introduce some housekeeping items. Please feel free to ask questions throughout. You can just use the chat, and myself or Chinmaya will be filtering and feeding the questions to the speakers. And I believe that's it for now. So I'll give the mic over to Vern, who I believe has the next slide here.
Great, thanks, Jen. And yeah, I'd like to extend my warm welcome to everybody that's online. And please do chip in with questions if you have them as we go along. Just to set some scene about Cyclica, I mean, Jen's given you a little bit of context on, what the Molecule to Medicine series is trying to do and what we're trying to do as a company to give back in the education space. But a little bit about Cyclica in general. It's a company that's about 8 years old. It's really been founded and funded to actually tackle some important things in terms of the underlying philosophy of Cyclica, which is to really understand how molecules bind to proteins. And we use the word polypharmacology. So a molecule or a drug is normally aimed at a specific place, and we'll get into that as we go through the slides. That'll become clearer. But also, when a molecule enters the body, or when you take a tablet or have an injection, it obviously flows through the body and goes into, through your bloodstream into different parts of your body, and also can go into different places. So the underlying philosophy of Cyclica was really to try and understand where the molecule goes and try and de-risk that. So try and understand the polypharmacology of medicines that people might be taking or drugs that they might be developing. And that's the underlying uniqueness of Cyclica. And it's something that is unique in the way in which we've designed our science and have our deep learning and machine learning engines apply that knowledge to our platforms of ligand express and ligand design, so that we can try and de-risk, but also design medicines with less risk, as by understanding how they go through the body and how they might interact with different proteins that we have within our human system. So, as I said, we've been going for about 8 years. We've also graciously been acknowledged by a number of independent organizations, which rank us as one of the leading companies in the drug discovery space. And really today, what we're trying to do with Andrew here is to try and just have a conversation between the two of us and to try and explain, give a little bit of insight and overview of what you saw is the Molecule to Medicine series, and then raise some points and just put down some markers, which if you're gracious enough to stay with us through the whole course of the 8 episodes, then hopefully will become expanded upon in each of those episodes as we walk through the program. So I don't have control of the slides, which is good because that means it'll get done properly. So Jen, if we can have the next slide, please. Okay, so we're going to just start with an overview of the discovery process, and I'll hand over to Andy now just to talk us through the first slide.
Hello, everyone. Thank you to Cyclica and Vern for inviting me to speak today, and thank you, obviously, everyone who's listening. As this slide indicates, I'm really going to give you a very high-level, 20,000 feet view of the drug discovery process. I can only do that because each molecule really makes its own journey. The path from an idea to a marketed drug is dependent on a lot of factors, some of which we'll discuss today, and obviously you'll get a lot more detail in the subsequent episodes of that. But it will depend on the disease, on the type of molecule that's being pursued. It will also be affected by changes in outside science, by changes in regulatory environments, or indeed by companies changing their own philosophy and practices. So as I say, each drug makes its own journey, but I'll try and give you a kind of generic, high-level view of what that process looks like. Now, I've asterisked up here drug discovery because one of my particular personal peeves is that you discover laws of nature, you discover planets and continents, because those are things that already exist. In fact, most modern drugs are inventions. Somebody actually had to have an idea, and somebody had to turn that idea into practice. That idea almost always starts with a disease. So we're going to talk about what a disease is a little bit, and how that may have changed. Our concept of what a disease is may have changed. We're going to talk about what makes good drug targets and how we can modulate them. How can we affect them so that we can affect the course of a disease? And then finally, we're going to talk in a bit more detail about what actually makes a drug. Vern has already alluded to the fact that medicines have to go through a long journey to get to the target to affect the disease in a patient. And we're going to talk a little bit about some elements of that journey and what properties a molecule has to have to be successful. So if we could please move on to the next slide. We'll start off with what causes a disease. I think everyone has an idea of what a disease is. It's something that makes you feel less well than you might otherwise be. Something that makes you feel uneasy or dis-easy, which is where the name comes from, disease. What causes disease? Well, some diseases are caused by primarily genetics. Some are caused by environmental factors, or toxins that might be in the environment that you've been exposed to. And some are as a result of changes in the way that your body is working, usually a breakdown or a failure of some natural processes, often through trauma or maybe old age, or in some cases autoimmune diseases. As you age, you are more likely to see these kind of degradations of your natural processes, which is one of the reasons why as you get older, you tend to suffer more from disease. But diseases can also have a lot of different factors. They may be partially genetic, partially environmental, as well as partially due to changes in an individual's natural body chemistry. And that makes it very difficult sometimes to come up with an actual cause for a specific disease. So we often end up characterizing disease by a set of symptoms rather than by whatever the underlying factor might be, because it's easier to identify symptoms sometimes than underlying causes. And as a result, we often also then treat to relieve those symptoms rather than addressing the underlying issue. But more modern medicine, as we've understood more about the processes involved in the development of diseases, we've tended more recently to give a greater emphasis on that underlying cause, looking to see what that factor is or factors are that give rise to disease.
Yeah, and Andy, that's just the point actually that's really important, because we are treating symptoms, we're not curing. Okay, and I think,
on the whole, occasionally, when we really do well and we're lucky, we can cure.
But it's majority of the time, we're really trying to treat the symptoms, to slow down progress, to try and basically reverse, if we can, what's going on in the body, to try and help people in general just overcome whatever the affliction is that they currently have. And it's rare that we can cure. Science and technology is moving forward, so there are other modalities and ways of trying to do that now, which are coming to the fore. But again, it's science and we're learning as we're going. So I think in the future, that's likely to be more the case of curing rather than some symptoms. But at the moment, it is definitely trying to treat the symptoms. So for cancer, you're trying to stop tumor spreading. You're not, the underlying cause of why the tumor's formed is not necessarily where the medicine's actually, or the treatment's actually being aimed at.
That's right. But as you do say, and I think it's worth being optimistic about this, particularly as we understand more about the genetic drivers and the key genetic drivers, and we have the options of gene therapy. We will move, I am sure, over the next few decades, much more towards treating those underlying causes. But you're right. Right now, and historically, drug invention, drug discovery has been focused more on symptoms than cure, simply because that's the way that we have understood diseases. sorry, before we move on, diseases can be short-term. You get a cold, lasts for a week or two, and it's gone, or they can be long-term. Okay, and again, whether it's a short-term disease or a long-term disease will affect the path a molecule follows through the drug discovery process. And I also just wanted to say before we move on that our concept of what is a disease changes. So the kind of things that we might be treating now may not have been considered to be diseases a long time ago. So I think, for instance, it's very much now that we would consider something like addiction or alcoholism to be a disease and something worthy of pharmaceutical or other intervention. Whereas 30 or 40 years ago, it would just have been seen as a character failure. But we move on and our definition of disease progresses, and that also impacts how we do drug discovery. So sorry, now we could please move on to the next slide. Thank you. So the first part of any exercise in treating a disease is to understand the disease. And that goes into this topic of target identification. Most of the drug targets that we work with are proteins, such as enzymes or receptors. And what we try and do is to alter their function. We try to make them either do more work, less work, or sometimes block them altogether so that their function is stopped, with the expectation that changing that function will produce downstream effects that we can measure in patients, in clinical trials, and that will be of benefit to those patients. That's kind of the overall process that we are aiming at as pharmaceutical scientists. We are looking, or bearing in mind always, that it's an ultimate benefit to the patients that's got to be the driver. Nobody will pay for a medicine which doesn't give them any obvious benefit, however wonderful the science behind it might be. There's some really nice drug discovery processes that have been gone through, which in the end gave no benefit to the patients and therefore didn't make new medicines. The science was great. There's nothing wrong with the science, but if it's not of benefit to patients, then it's not been a successful drug invention exercise.
Yeah, and if we just focus on a couple of things from this particular slide, which always gets to me. So proteins, there's an estimate about 20 to 80,000 human proteins in the body. Okay, and from what we know is they are pretty much, most of them are interconnected. So although you might identify a protein as a target, it actually sits on a pathway with other proteins and other things, such that actually when you impact one protein, it can have sort of impact elsewhere on other proteins along that particular pathway. So the complexity of what tries to be done in discovering or inventing a medicine is actually quite high, because nothing is independent. And the other thing is that, going back to the benefit piece, is when you take something, because back to your point, actually, it's not a discovery because it didn't exist before, so it's an invention. Actually, it's the risk of taking that particular medicine to the patient compared to the benefit that the patient gets. So that risk benefit is actually something which is really at the front of mind of everybody when they're looking to discover and invent new medicines, so that you can actually understand, is the benefit outweighing the risk? And that really does depend on the type of ailment that you have. So the risk benefit for a headache, as an example, is, okay, you have a headache, there is a pain with that. Some people don't want to take any medication for that, but if you're taking something, you will want it to be, tuned in to the kind of the headache that you have. But if you have a very serious disease where, there are very little treatments for you, you will take a higher risk, or you will accept a higher risk in the treatment, that it may have some side effects, which, you're aware of, but actually you're willing to live with those side effects because it has the potential to slow down or reverse, that particular disease that you have. So that risk benefit is really something which is at play throughout all of the inventions of medicines as you go through, and that changes as you get, as you get more knowledge on the disease and obviously on the patient population, because younger children compared to older people, the risk there is different as well. So therefore, there's a lot of factors that we've got to take into account when we're doing these inventions.
Absolutely right, Vern. And if we go back to the previous slide here, we can talk about it a little more. And the other thing which one always has to bear in mind is what else is out there to treat that disease. So even if you have a serious disease, if there's something already on the market which is pretty reasonable and has a good safety profile, that's your benchmark for an improvement. People are not going to take extra risks, exactly as you say, with a new medicine just because it's new, unless it provides extra benefits. So that has an impact on the clinical path. Right back to what I said at the beginning, the type of disease, the risk profile of the molecule that you're interested in, and the risk profile of competitors all factors into what is the process I need to follow to get from here to something of benefit to the patients downstream. We need to go back one more slide, please. A lot of what we just talked about actually is encapsulated on here. Successful invention of a drug requires two things. Essentially, it requires a target which is relevant to that disease. And that's not always an easy thing to establish, as we've already said. Sometimes there are multiple factors involved in the development of a disease, and identifying a target or even targets, go back to something that Vern talked about, polypharmacology, molecules that interact with more than one target. Identifying those targets is not trivial by any stretch of the imagination. And then there's this thing here, which I put in brackets, which is exactly what Vern was just talking about. This target must also be safe. And safe is a relative term here. It's not an absolute term. The complexity, as Vern said, of the human body is amazing. It's fantastic how resistant we are actually to disease, because we very often have multiple layers of defense, multiple layers of control over the processes in our body, and very often 2, 3, or even more of those need to break down or be interfered with in order to affect an overall function. And therefore, you're not going to get something which is 100% safe, that has no risk at all. Equally, as you said rightly, Vern, when we come up with a ligand for that target, which is going to be the molecule that we're going to progress to a medicine, it also has to have a safety profile. But again, we can't know everything about that, every individual and about every molecule. And therefore, safe here is also a relative term. And that brings in the concept, which I think he tried to introduce there, of risk benefit. Well, he did introduce there, sorry, of risk benefit, that there must be a demonstrable benefit to the patient which exceeds the risk. And that risk benefit is variable, again, depending on the disease, exactly as you said. So it is clear, although I'll talk about it a bit later on, that the clinical trial process, the risk for someone who has a really aggressive cancer, which is going to kill them in weeks or months, you can take more risks, or those individuals are willing to take more risks than someone with, as you say, a mild headache, which is not to belittle the fact that people, for instance, who have migraines have a very severe pain and dysfunction issue, which does need treatment. I think one important point is many clinical trials fail. It is a risky process. And if they fail, but you identify that your molecule was safe, you reached the exposure that you wanted to in people, in patients, and that the target was just not critical to that disease, that's not a bad thing. You have learned something. That is an advancement in science. No one else then need go back and make another ligand for that target. What is a real killer when it comes to deriving value from clinical trials are those which fail, and then you don't know if it's because your ligand wasn't safe enough or because your target was not involved in that disease. That is a very expensive, that is a real failure, I think, in the clinical process or in the scientific process that led to it.
Yeah.
So that's the situation to avoid. And that's why I think it's very important early on in the process of drug invention, which is the part of the process that I've been most involved with during my career, to take your time and make sure that you have the best possible information on your target or targets, and that you've chosen a ligand which is capable of giving good exposure in humans and testing that target in a clinical trial.
Yeah, and I think it's important to emphasize that actually the discovery, because what you invent as a ligand or what you discover as a target is actually then what you carry through to all the way through to a medicine. Yeah. And so therefore, whatever baggage, if that's the right word, or whatever things, the liabilities that you're carrying on the molecule, for instance, are things that you're going to have to manage as you go forward. So spending time at the beginning actually enables you to kind of speed up a bit later, or actually have less issues later on. So although it feels sometimes it can take, and I know when I was working at GSK, it sometimes took us 3 to 4 years to get to a molecule that we wish to progress to the next phase of preclinical, so before the clinic studies, and it took us 2,000 to 3,000 molecules to get there. That was time that was probably worth spending, because actually later on you have more knowledge of the molecule, and it should allow you to work through the next phases that much more easily, should I say. So yeah, absolutely. It is absolutely worth spending the time up front in this case.
Indeed, I've been a medicinal chemist for all of my career, and however good a medicinal chemist you are, if you start off on a target which is not related to the disease that you're supposed to be looking at, there's nothing you can do. However brilliant your molecule is, it's still going to fail in the clinic. So right. Absolutely, I think it's probably still an area that we need more patience in identifying those good targets. And that is complicated because not everyone is the same. We all have different environments. We all have different genetics. And so we don't all respond to diseases in the same way. And going back to what I said before, we tend to categorize diseases by symptoms. But therefore, that sometimes means you get people included in a clinical trial or in a process who don't have the exact disease that you're interested in, or sometimes other people get excluded and you kind of miss things out. So identifying those targets is absolutely critical, very early on. It really sets the scene.
That's where the genetics now that's where the advances in genetics now can help, because I think you can look at the genetics of individuals and then actually tailor potentially or understand those with similar genetics may have similar sort of responses to the disease themselves. And that, I know we'll probably touch on that later on, but actually the advent of the understanding of genetics is now playing a very much larger part in helping us understand which proteins, why certain patients do respond to certain treatments and others that supposedly have the same disease don't. So there's a lot of science which is moving forward. So trying to just bring a more optimistic approach than 3 to 4 years and 2,000 to 3,000 molecules. But there is some greater understanding now, which is helping to push things further forward.
Yes, I think the genetics is clearly playing a role in identifying targets, and perhaps more importantly at this stage, identifying those pathways and helping us to choose which points on those pathways to intervene at, which are going to have an impact on most patients. But as you rightly say, it also enables us sometimes to go back and say, well, why only did 30% of the people in clinical trial A respond? And we can identify actually there's a whole subset of people who have one particular pathway which is dysfunctional in a disease, and there's another set of people who ostensibly have the same disease but have a different pathway. Different mechanisms which are failing and therefore won't respond to that first therapy. I mean, historically, the way we tried to predict targets was as much based on cell and animal models as anything else before the real rise of genomics and genetics. But there are some diseases, and I mentioned here one of my own, I won't call it favorite disease, but disease I worked on for a long time, Alzheimer's disease, where there are no models. You can't use animal models to predict how well your target is going to behave, which is one of the reasons why diseases like Alzheimer's have been very slow in delivering new therapies or effective therapies to patients. And again, genetics is now playing a big part in the kinds of targets which are entering the clinic now.
Andrew, I've just received a question from the audience. Do you mind if I
Please ask
it?
away.
Okay. So, question for Andrew. For complex diseases like neurodegenerative, multiple targets are often implicated in such disease. How does one validate which target to pursue? Can you give an example of when conventional validation of a target was proven to be wrong and what was learned through that?
That's a really good question. So neurodegeneration is a very complex area, as I'm guessing the questioner knows. In fact, it's one of the areas where it's even difficult to define the disease. So again, if I think of Alzheimer's disease, the way that it is now defined is as much based on biomarkers of amyloid and tau, which are proteins which are overexpressed in the brain of these patients, as anything else. So there's almost an assumption that that is an underlying mechanism of the disease. It's not proven. It's just the way that we define it has led to those being accepted as targets. But I think it's still very open to question as to whether amyloid is indeed a key component. Now, there has been an approval, as some of you will know, this year of a medicine targeting amyloid by the FDA as a therapy for Alzheimer's. It will be interesting to see, as that medicine is prescribed, how it develops. But up until that point, I would have said the answer to your question was that genetics favored amyloid as a key component. Pharmacology, certainly post-mortem, pharmacology and post-mortem analysis of Alzheimer's patients also favored amyloid as a target. And yet up until that point, many amyloid therapies had failed in the clinic. Now, there may be other reasons for that, not related directly to the target, but it's an example of where things get complicated and also where we kind of make assumptions based on the symptoms, or in this case, a biomarker, as to what causes the disease, which may or may not be right. So I would say that amyloid therapy has been the mainstay of Alzheimer's treatment for 20, 30 or more years, and it's still unproven. Hopefully that answered the question. It was a long answer to what was probably quite a short question.
Great, thank you. We have another question. How has the emergence of data relating to disease biomarkers and or treatment emergent biomarkers affected target identification? That wasn't specific to Andrew or Vern, so either one of you are able to answer.
I'll give it a go at the start. And I think, again, it depends on this question of whether or not you have a useful animal model in terms of the drug discovery or drug invention process. If you have a model which is predictive of the human disease and you have a translatable biomarker in that model which can be translated into patients, then that's a great situation to be in. And there are examples where that is the case. And there are therefore examples where progress has been made with some diseases based on biomarker strategy. I go back again to what I just said in terms of the biomarkers for amyloid. They are kind of defined as biomarkers that tell you that you have amyloid. As of yet, we don't really have that relationship between understanding of those biomarkers and the actual causes of disease. So it's a mixed bag, I would say.
I'd add to that, Andrew. I think people have. I mean, what we'd really like to be able to do is put the patient at the front in discovery. Okay, so what we're doing in discovery is trying to identify those things which, the target or what it could be that we could be impacting to improve the outcome in terms of a medicine for the patient. So what now countries, actually national countries, are actually doing, and the UK, which is where Andrew and I are based, have done is they have what's called the UK Biobank, which is a collection of patient data, and it's different types of data from the same patients, and I can't remember, it's like 500,000 people, I think. There's lots of people in that collection, and these are data that are collected over time, and that's been made available to researchers and they are mining that data to find the opportunity to go back to one of the points we've already made, where you can see that some patients have, a particular pathway in a disease, and for that same disease, a different set of patients have very much a different pathway, which means that actually when you set up a clinical trial in the past, you would look for people that had that disease, not really understanding necessarily the pathways to the disease that each of them sort of suffer or have. Whereas now it's possible to maybe identify patient groups which are more similar, and therefore we can start to see if we can treat or try to help patients in certain subcategories of an overall disease that would be trying to be treated. So the Alzheimer's, the amyloids, certain might be one of them. There might be other, certainly are other targets that will be impacting from an Alzheimer's perspective. So data's helping a lot. And I think people are waking up and countries are waking up. And there's some really good data sets which have been well curated. So they are, they are fairly easy to kind of interrogate, which now are becoming available to researchers across the world. And there are companies like 23andMe where people sign up to have their genetics, so they can understand the genetic profile in a more social way, which now have a great repository of genetic information on people. And that information now is being mined to see if it can be used to identify new targets for potential sort of medicine invention. So I'd say it makes a big difference. And I think it'll just increase as time goes and these things become available.
Absolutely right, Vern. I think it's worth, I'm going to reiterate something that you said.
Sure.
One of the big advances that is being made and will continue to grow, I think, is the selection of patients in a clinical trial based on our understanding of the target that's being examined, and also those individuals' genome, that we can stratify diseases more readily now than ever before, and so the likelihood is we will be able to find effective therapies for certain subsets of disease. And because I've talked about it before, that for instance in Parkinson's disease there are several different cohorts across the world, certainly several here in the UK, and being able to stratify those patient populations by their genetic profiling or by the changes that have led to them having Parkinson's disease is really a big opportunity when it comes to the development of new and targeted therapies.
Yeah.
Fantastic. So we do have a couple other questions, but I am concerned about time. So should we move on to?
I'll try and get through this as quickly as I can so we can get back to the questions. I think the questions are really important, but
do you want me to, they're mostly with regard to target ID. So the previous slide.
Let's come back to them and see if maybe we may answer some on the way. Just do a couple of slides and then maybe come back to them. So we've talked about target identification. So the other arm of drug invention is ligand identification. And historically, actually most 30, 40, 50 years ago, most ligand identification would have been done through this process of phenotypic screen at the bottom. So using animal models, using cell models, and identifying changes in function that led to some measurable change in those cells or animals that we thought was related to a disease. More recently, we have tended to develop new targets based on high throughput screening, so the screening of hundreds of thousands, maybe sometimes even millions of compounds, and identifying a ligand for a particular target that way. And even more recently, and obviously a big focus for the kinds of technologies that Cyclica are interested in, but I have to say other in silico approaches are available.
Absolutely.
Is the use of in silico screening, virtual screening of compounds against a 3D structure. So 3D structures for many targets, proteins, are almost routinely available now, and predictions are being improved on an almost daily basis, even for structures where we don't actually have an X-ray or some other way of measuring the 3D structure. So that only going to advance, I think, that more and more often we will generate our starting points through the use of in silico technologies. And in conjunction with that is something that we'll come on to, and maybe we'll have to race through it a bit so we won't get as much time on it as I would like. But in conjunction with that, we'll also be able to predict the properties of those compounds and those hits better as well, which should all save time and money and effort when it comes to identifying those good ligands which are suitable to go into clinical trials.
Yeah. And just picking up, and only because it's been in the news fairly recently, is the big announcement from DeepMind and the MBL that, you know, the AlphaFold sort of announcement that they made, and those technologies are there really to help predict the structures of proteins from just their sequence. And that's something that we're already starting to incorporate in our work to help us sort of improve our efforts in actually trying to design chemistry towards certain protein targets. So yeah, no, absolutely, science moves forward.
So however you generate your starting point, you're going to have to optimize it almost certainly. There are very few examples where someone has come up with a molecule which has gone to market straight from some kind of screening exercise, unless it was already a pre-approved medicine. So there are things that you get a target, you'll get a ligand for that target, and there'll be things that need to be improved, maybe the potency or the selectivity and some other issues that we'll come on to in a slide or two's time. Traditionally, what one has done is to run one's SAR, structure-activity relationship, through a kind of flow scheme, where you'll make predictions here on the left, the in silico predictions, you'll make some compounds, and then you'll test those compounds. Usually a flow scheme is ordered by the highest capacity assays run on the left-hand side here. So that's what most of your compounds go through, those high capacity assays, which normally will involve affinity for your primary target, for instance, as an example. And then the more expensive and lower throughput assays tend to be the in vivo ones, will be run later on fewer compounds, because that's more expensive. And obviously, it's not ethical to test unsuitable molecules in animals anyway. Sometimes you'll have specific issues. Maybe there'll be some issues about whether the molecule can actually survive being in the stomach of the animal, or cross from that stomach through into the bloodstream, that you may need to bring assays which normally would be run later on forward. But generally speaking, you tend to run them in this fashion of highest throughput assays on the left, and therefore testing most of your compounds, and lower throughput ones only on the most advanced molecules, the ones with the greatest prospect of actually being clinically suitable. So if we move on, please. I mentioned this before, so did Vern. A medicine really does make an epic journey to get from being a pill or an injection or whatever it might happen to be to your bloodstream and then into the target. So if you take a pill in your mouth, it's going to pass down into your stomach. It's going to be into the intestine. So the stomach has a very acidic environment. The intestine has a gradually more alkaline environment. It gets absorbed from the intestine through the so-called portal vein, and then into the liver. And the liver is a powerhouse of metabolism. It really wants to change everything that comes through it. It essentially is a detoxification organ that tries to make sure that nothing poisonous gets into your bloodstream, which is great for protecting you from disease. But when you have a ligand which you're hoping will do some benefit to that patient, it's not so good, because it's constantly trying to change that molecule into something else. So even to get that far, you require a ligand which has great stability across a range of pH and different environments. Then it will get to your target. And of course, some targets are very difficult to get to. We'll talk a little bit more about that in the next slide or so. But the important take-home message from this slide is that there is a very small subset of potent compounds in your SAR, in your structure-activity relationship, which will have the right properties to have the kind of stability and the kind of permeability to get where they need to be a medicine. And laying on top of that, which we haven't really talked about much at all with our molecules, is they've got to be safe, or safe in quotation marks. They've got to have that reasonable safety profile as well. So that flow scheme that we had on the previous slide is a way of triaging down to get to those ligands which are most suitable to go into clinical trials. And I think on the next slide, we'll just get some more background. I think I'll actually, Vern probably talked a little bit about this from his experience at GSK. And I have to say, it's been my experience as well. This is a lengthy and iterative process. It can be shortened if you start with a high quality hit. But generally speaking, it's going to take hundreds, if not thousands of compounds, and it's going to take 18 months would be very fast for this kind of process. Because we're optimizing for multiple issues, and we'll talk on the next slide about what those issues may be, but it's also, as you fix one process, and again, I think Vern alluded to this earlier, you often end up unfixing something else, or breaking, I suppose is probably the right word there, breaking something else. So you fix permeability, you ruin solubility. You fix solubility, you ruin metabolic stability. It's very hard to get all of those things right in one molecule.
And in essence, what you come out with is kind of the best balance of all of those things.
Yes.
So that you actually try and get the best balance of that. And I think the other thing that's important to just recognize, because you've got your active ligand, but then, as you said, you've got to deliver it to the right place in the human body, and the formulation. So how you put that active molecule together with other components, or excipients as they're called, to make, for instance, as a tablet, is really, it's a phenomenal set of sciences just to do that, to make sure that actually the molecule gets to the right place and survives the low pH in the stomach, the high pH in the gut, and then moves through to where you need it to be and protect it from the liver. That science has to be done to protect your ligand, but also it's got to work every time the same way. So the reproducibility of making a tablet is actually a phenomenal challenge as well to get a medicine, as it is for an injection, as it is for other types of approaches in how you deliver your ligand in. So not only once you've found it, actually to get it to the right place actually makes it's a really impressive job when it actually can make it work.
Absolutely. Pharmaceutical science, as is essentially what you're talking about there, is a real science of worthy of a whole kind of seminar itself. Yeah. Well, worthy of several seminars actually. It really is a complex process. What goes into a pill, as you alluded to, is phenomenal. And we'll talk about reproducibility, but yeah, essentially you want to be able to get the same exposure across multiple patients. I mean, this is the other thing. We talk about reproducibility, but of course I can take a pill and get a different level of exposure to Vern or Jennifer or whoever else, simply because I'm a different person. We just don't know how we're going to respond until you take it. There's always that risk. So if you just go back, keep on this slide for a second. So the safety is key. There's some other characteristics here which we're really going to have no time at all to go into, but I'm going to put in a plug for aqueous solubility. Is often an underestimated characteristic that is really useful to have in a drug, because it speaks to this question of reproducibility, because most of your processes in your body carry on essentially in aqueous solution. If your compound is in solution in water, that helps. If it's not very soluble in water and it's precipitating out all the time, that's where you get this lack of reproducibility. But yes, we need things like bioavailability. All of these things I'm sure will be covered later on in much more detail. But you need to get your compound into your animal, into your patient. You need to get it to the target, and it needs to stay there long enough to have an effect. Sometimes an effect is very rapid when you interact with the target, but sometimes you just have to stay there all the time. For many chronic diseases, you have to have your molecule on that target 24 hours a day, 7 days a week, 365 days a year. And that is not a trivial achievement. Let's, I know we're running out of time now, so if we move on, there's no time to go through that. We've already spoken to the fact that in silico tools are very useful now for identifying novel targets and novel ligands, but I also mentioned that actually the advances in our ability to screen large databases of compounds and large data sets has greatly improved our ability to find molecules that have the right property space. So for me, for instance, having worked in neurodegeneration for a long time, one of the big characteristics the molecule needs to have is to be able to get into the brain. And your brain is another one of these wonderful things that evolution's provided us with, is strongly protected by the blood-brain barrier. So even once the liver has detoxified anything, if anything gets through, the blood-brain barrier, for the most part, keeps things out, including your medicines. So, but there are properties that compounds can have which will allow you to pass through, and being able to calculate those, being able to identify whether your targets and your molecules have those properties has been a real advance, I would say, over the past decade or so. And again, hopefully will lead to real improvements in our ability to treat some of these central diseases. But it's still not perfect. It still can take a lot of time and a lot of effort to get to where we need to be. Once we've identified a candidate, which in our flow scheme gets to the end and has demonstrated efficacy in whatever animal or cellular models we have chosen to define efficacy in, and has demonstrated a safety profile. So normally that means several days or weeks of safety in a couple of animals, usually a rodent and a dog, that gives us sufficient confidence that there's a risk, and that the risk-benefit ratio is in favor of taking this molecule into people. So that's a big step. Many projects don't get that far. But obviously, putting a molecule into patients, even at a low dose, which is where we start, and in healthy volunteers, which is usually where we start, it is a risk. And so you need to be pretty sure that you've got the right compound, and that compound, as Vern alluded to earlier, is of high enough purity and controlled well enough that it's safe enough to go into people. So you know, very often, for instance, one of the big characteristics that you'll need is that you'll have a crystalline molecule, but you need it to crystallize in the same form and the same particle size pretty much every time. And that's one of those elements of pharmaceutical science, which is a black box to me, but which the people involved in it are remarkably brilliant at, in my experience. And that leads, if you can control that form, to the reproducibility, which means that you can go into the clinic. In the clinic, obviously, this is the process in patients, eventually, by which you demonstrate safety and efficacy in such a way that you can convince a regulatory authority to license a drug for use. That's the end game that you want from these clinical trials. This is, I think, the last slide is coming up now. The process, which, and again, this is a very generic process. If we move on to the next slide, please. It's a very generic process. If you're dealing in oncology, it is slightly different, but I'll stick with this generic one. The phase 1 trials tend to be low dose, healthy volunteers, and relatively small numbers. And the focus here is on establishing the safety of the compound. After single dosing, that will be the very first set of trials, and then repeat dosing. So this gives you confidence that the molecule is not so unsafe that you'll never be able to treat patients with it. You also can generate data on how well exposed the compound is in people. So to figure up to this point, all your data has at best been generated in animals. You'll get to see how well that data translates into people after phase 1. Phase 2 trials tend to be a bit longer. They may go on for weeks, months. And they tend to be in larger numbers of people. And this is the point at which you really test your compounds in patients. So there's a continued emphasis on safety here, but you can also get efficacy data. And this is where we provide, hopefully, a proof of concept. This is the data point which speaks to something I mentioned very early on. Is your target related to the disease that you're interested in? If you can get a positive result in phase 2, you can say to yourself, yes, my target and this disease were related. If you don't get a positive result in phase 2, and you haven't got the exposure you wanted, then you can't answer that question. That's what I would consider to be a failure. But obviously, if you don't see efficacy and you did get exposure, you still learned something as a result of this. You've learned that that's not a good target for this disease. So not a failure, but obviously not useful for patients. Finally, if you get a positive result there, you can move into the phase 3 registration trials. These tend to be much larger. They can be thousands of patients, and they can go on for several years, particularly for chronic diseases. And these tend to be the pivotal trials that organizations like the FDA, the regulatory authorities across the world will use to establish the long-term safety and efficacy of a drug, and whether it's approvable or not, whether there's a benefit to the patients which outweighs any risk associated with that medicine. It is also worth pointing out there is actually a phase 4. Once a drug has been approved, we do continue monitoring the safety of that medicine in the patients that continue to take it. Because, of course, even in a large trial with thousands of patients, that gives you no idea of how a drug is going to behave in the millions of patients who may end up taking it. Yes. Because, as I keep saying, everyone is different. There may well be some very rare side effects that we need to know about. And we may, using that genetic data we talked about, be able to say, actually, these patients should not take this drug for this reason, because the risk is suddenly much greater. So that ongoing safety process is absolutely critical, and really never stops as long as a medicine is being taken by patients. Which
is why we get those big long leaflets inside your medicine boxes, which tells you all the things that you could be potentially feeling as potential side effects, which you will have discovered along the road, plus some contact numbers about who to get in touch with if you feel some of those. Because it really is only when you get into the millions that you actually get a really solid picture of how whether the risk benefit is there, or what things are occurring to those patients that maybe you hadn't quite identified even in the phase 3 trials, which, like you say, can run into thousands over a period of years. So safety is constant monitoring by companies that have medicines on the market. And in fact, the major thing that companies are looking to try and establish is how safe they are, and then the benefit that can bring. So it tends to be that way around, rather than, oh, this is really good, yeah, but it's got a bit of liability. It tends to be, let's work out how safe this thing is, and then see if we can get the efficacy as well.
If nothing else, I would encourage everyone who's listening or watching, if you do take a medicine and you do have some adverse event, please report it.
Report it.
It really is important that we do so.
Yeah. Okay, Andrew, that's been great. Thanks for that. That's good.
That's been fantastic. I think the next slide is simply do we have any questions? And as we didn't get to some of the earlier ones, I'm hoping we can sneak one in now before the end of the session, if that's all right. Can I ask one from the audience? So this lovely person says, hi, I've been really enjoying the topic. I have a question regarding the target selection in the process of drug discovery. I worked on several small molecules targeting P53, but the problem with these molecules is some targets wild type, or some mutant, or some both. How can we approach to be more focused in terms of selecting specific targets?
Yeah, I've never worked on that target myself, but it's a very good question, very good, in general terms, that hopefully, or I would expect that the advances in genomics will be really useful here. But it speaks to the fact that we are genetically diverse, and an understanding of disease and disease mechanisms needs to allow for that genetic diversity. There clearly are targets which are very good for some diseases in some subsets of the patient population, and there are mutations in that target which can lead to disease, but there are equally mutations in that target which can prevent the medicine working. And really, the answer to the question is, as far as I can tell, is we just need to improve our understanding of disease and the relationship between that disease, and in particular in genetics. I think, historically, we tended to approach disease as a one-size-fits-all. We assumed that people were genetically homogeneous on the whole. People are not. And so there were a lot of work done on a lot of targets, I think, where there was never going to be a substantial, broad impact in a clinical trial that would demonstrate benefit. Maybe if we looked at some of those targets and focused, as I said earlier, and actually identified a subset with a particular genetic profile, you would be able to see that benefit. But against a homogeneous a heterogeneous patient population, you were never going to see it. Now, think about many medicines. If I go back to what I was talking about earlier, it's been used to try and treat Parkinson's disease. But if you're treating Parkinson's disease with a medicine which affects target A, but you're treating it where 70% of your patients have no problems with target A, it's target B that they have an issue with, your signal in that clinical trial is going to be swamped by the 70% who are never going to show any benefit. You won't be able to see it. And so that identifying of the right patient for the right medicine is absolutely critical.
Yeah.
Well said, Andrew. Thank you so much. I think that's an excellent place to close. And thank you both, Vern and Andrew, for delivering really, really interesting and insightful presentation. We had loads of questions that we couldn't get to. So we will reach out to these attendees, and perhaps they could follow up with you by email, but we'll keep you posted on that. For everyone still here, this recording will be available on YouTube shortly following this. And stay tuned for October 6th at noon will be episode 2, and we will specifically get into the nitty-gritty details of target ID, which evidently generated the most questions within this topic today. So we're looking forward to that. So please sign up. You can do so on our website and cyclicarx.com. And thank you everyone for joining, and thanks again to our speakers. Have a nice afternoon or evening, everyone.
Thanks for joining us.
Thanks a lot.
Okay, all good.
Are we still live?
Yeah.